We're here at the Cahaba River, just right here pretty much in Birmingham. So we're just downstream of the Highway 280 bridge, and we're conducting a quantitative mussel survey. So freshwater mussels, as I mentioned before, are North America's most imperiled faunal group, if not the most imperiled group in the world. We're also conducting sampling related to the environment that they live in. So we'll collect water chemistry samples to analyze.
There's two ways to look at what mussels are, like in a site or in a river. One way to do it is doing qualitative, so basically you go there, snorkel around and see what's there. And the other way is doing it quantitatively, and that allows you to actually make population estimates and know like the density of mussels that there's in a site. We're using a flow meter that measures water velocity. Today we're measuring it at the substrate, so where the mussels are, but it actually can measure like discharge at different points in the water channel.
We have folks from the Alabama Department of Conservation and Natural Resources. They're going behind with a 1/4 meter squared quadrat, and they're digging up all the sediment, excavating it, placing it in a bag. And that allows us to get, an estimate of how many mussels are in that particular area. And then we go through and we sieve all of that material and find each individual mussel. And so that sort of data allows us to estimate the species richness of this site, so examine how many species there are. We can also estimate things like density. So given that we are sampling a set area multiple times within this particular reach, we estimate the number of mussels per meter squared. That gives us a lot of very useful information regarding, habitat preferences. So we're making sure all of our environmental data corresponds to our mussel data. Some mussels prefer living in soft substrates, softer sediments, and so we can correlate the data that we're collecting with habitat preferences for a particular species.
I'm really interested in how they change, like how the shape varies. And usually to study that, you take pictures of the organism you're studying. And for example, for fish, they do it a lot. They just take pictures, but that just gives you like a 2D perspective of it. And with mussels, depth is really important on the mussel. And with the 3D scanning, what you see is like all the dimensions of it, right? So what I'm doing is I'm actually working with the anthropology department. They have a really nice 3D scan. And I go there and like take these like scans of mussels. And what I'm doing is comparing the shapes in the different rivers, in the different sites, and see how they vary based on like environmental characteristics. And then we have another person doing population genetics in the mussels. So we'll see if those differences, the variation is actually from like just environmental factors like water velocity, what we were measuring, or substrate, or actually there's like genetic differences that are making these populations different. So I'm really interested in studying mussels. I got my master's at Arkansas State studying mussels, freshwater mussels. So like Alabama is the best place to study mussels. They have the highest diversity, and also Carla is a great advisor for that. I really want to work with mussels, so that's like my goal. I'm open to do it in different ways. I really like what the guys in the agencies do because they're just involved in the field a lot. But I'm also enjoying working with undergrads here at the UA and showing them how to do some of the stuff that we do. So I'm getting more open to the possibility of becoming a professor one day, but I still have some time to think about it.